Hello! In today's video, we shall learn in detail about the secondary growth in plants. By the end of this video, you will have a clear understanding about the process of secondary growth in both dicot stem and dicot root with their differences, activity of both vascular cambium and cork cambium, formation of annual rings, differences between springwood and autumnwood, heartwood and sapwood. Let us begin with the introduction to secondary growth in plants. Growth is a fundamental characteristic of all living organisms, and it may be defined as a permanent and irreversible increase in the size, mass, or volume of a cell, an organ, or the entire organism in a given period of time. The two main types of growth in plants are primary growth and secondary growth. Increase in length of shoot and root of a plant as a result of cell division in apical meristem is referred to as primary growth. Increase in girth or thickness of stem or root of a plant as a result of cell division in lateral meristem is referred to as secondary growth. The formation of secondary vascular tissue from cambium or secondary growth is a characteristic feature of woody dicots and gymnosperms like conifers, but is generally absent in monocots as they lack a vascular cambium. Secondary growth helps to increase the plant's girth or thickness, provides structural support and protection, ensures conduction of water and nutrients to the entire plant body, and enhances longevity. Secondary growth takes place through the activity of two lateral meristems, namely vascular cambium and cork cambium. Vascular cambium consists of meristematic tissue that produces secondary xylem or wood and secondary phloem, while cork cambium produces a protective layer of bark on the outer side of the plant body and also helps in reducing water loss. Annual rings or growth rings are primarily seen in plants growing in temperate regions with distinct seasons as cambial activity fluctuates throughout the year. The wood formed in spring is called the springwood or early wood, and that formed in winter is called the autumn wood or late wood. A clear boundary can be seen between the late wood of one year and the early wood of the next year, resulting in the formation of the annual growth ring. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Next, let us learn about the process of secondary growth in a dicot stem. The meristematic cells or cambium located between the primary xylem and primary phloem of a vascular bundle is called intravascular cambium. They are known as primary meristem and are responsible for both primary and secondary growth in plants. The interfascicular cambium is located in the region between adjacent vascular bundles. They are known as secondary meristem and they originate by the dedifferentiation of parenchyma cells of the medullary rays. It joins with the intrafascicular cambium of adjacent vascular bundles, forming a complete ring known as cambium ring, which is responsible for the secondary growth in dicot plants and gymnosperms. The cambium ring consists of two types of cells: elongated spindle-shaped fusiform initials and shorter isodiametric ray initials. The fusiform initials divide to produce the secondary phloem towards the periphery and secondary xylem towards the center. Secondary phloem consists of sieve tubes, companion cells, phloem parenchyma, and bast fibers. Secondary xylem consists of scalariform and pitted vessels, tracheids, xylem parenchyma, and wood fibers. Secondary xylem increases more rapidly, accumulates and forms the bulk of the stem, which is known as wood. Primary xylem gradually approaches the center and forms a hard, compact mass. The primary phloem is present above the secondary phloem, but finally gets crushed as the secondary phloem accumulates and it becomes part of the bark. The ray initials divide to form a narrow band of parenchyma cells that become radially elongated and extend through the secondary xylem and secondary phloem. They are known as medullary rays. They help in radial transport of water, nutrients, provide mechanical support, and sometimes store nutrients. Next, let us understand the formation of annual rings. Annual rings or growth rings are seen in the cross-section of woody dicot stem. They are concentric layers of secondary xylem. Each annual ring consists of two distinct concentric growth rings, one of autumn wood and the other of spring wood. It is formed as a result of seasonal activity of the vascular cambium, especially in temperate regions. Spring wood or early wood is formed during favorable conditions in spring. The cambium is highly active and produces greater number of pitted vessels. The wood produced has larger cells with thinner walls and wider lumens. They are lighter in color and less dense. They constitute the major part of the annual ring. Autumn wood or late wood is formed towards the end of the growing season. The cambium becomes less active and forms narrow pitted vessels, tracheids, and wood fibers. The wood produced has smaller, thick-walled cells with narrow lumens. They are darker in color, denser, and stronger. A sharp line of demarcation is seen between the late wood of one growth season and early wood of next season. Both the types of wood appear together as concentric ring in transverse section of stem and is known as the annual ring or growth ring. Each annual ring corresponds to 1 year's growth. The study of annual ring is known as dendrochronology. Let us study the differences between spring wood and autumn wood. Spring wood is formed during the spring season, while autumn wood is formed during the winter season. Spring wood is otherwise known as early wood. Autumn wood is known as late wood. Spring wood occurs in the beginning of an annual ring, while autumn wood is formed towards the end of an annual ring. In spring wood, during the spring season, the cambial activity is high and produces abundant xylem elements. In autumn wood, during the winter season, the cambial activity slows down, leading to the formation of fewer number of xylem elements. The vessels formed in springwood have larger and wider lumen to make room for the increased water demand. The vessels formed in autumnwood has narrow lumens as the water requirement is reduced. The tracheary elements are less thickened in springwood, while the tracheary element in autumnwood are more thickened. Xylem fibers are fewer in number in springwood, while they are abundant in autumnwood. Springwood has lower density and autumnwood has higher density. Springwood are lighter in color, autumnwood are darker in color. Springwood constitutes the major part of annual ring. Autumnwood appears as a narrow strip in the annual ring. Next, let us learn about heartwood and sapwood. In an old tree trunk, the secondary xylem shows two distinct regions made up of two different types of wood, namely heartwood and sapwood. They differ in their physical properties, composition, and function. Both are essential for the survival of the tree. Heartwood is otherwise known as duramen. It is the dead, dense, central core of the tree trunk formed due to natural aging process of the tree. It is composed of dead, non-functional xylem cells, often plugged by tylosis. It provides mechanical strength and support, but do not take part in conduction of water and minerals. It is filled with organic compounds like tannins, resins, gums, etc. and is dark in color. As it is low in water or moisture content, it is naturally highly resistant to fungus and decay. Heartwood is hard and durable and is preferred for making furnitures. Sapwood is otherwise known as alburnum. It is the living outer part of a woody stem or tree trunk. It is light in color. Sapwood is physiologically active, consisting of xylem vessels and living parenchyma cells. The xylem vessels are responsible for the transport of water and minerals from the roots to the leaves. Parenchyma cells store food reserves. Sapwood is high in water content and lacks deposits. It is less durable and is susceptible to fungus and decay. Sapwood is soft, more elastic and flexible, and is used in the manufacture of plywood. Next, let us study the differences between heartwood and sapwood. Heartwood is also known as duramen. Sapwood is otherwise known as alburnum. Heartwood forms the central core of old stem, while sapwood forms the peripheral portion of old stem. Dead cells with lignified walls are present in heartwood. Sapwood contains living parenchyma cells and functional vascular tissues. Heartwood is the non-functional part of secondary xylem or wood, while sapwood is the functional part of secondary xylem. In heartwood, vessels are plugged by tylosis. In sapwood, vessels or tracheary elements are not plugged by tylosis. Deposition of organic compounds like tannins, resins, gums are seen in heartwood. In sapwood, tracheary elements do not possess any deposit in their lumen. Heartwood is darker in color, while sapwood is lighter in color. Heartwood has low moisture content. Moisture content is high in sapwood. Heartwood is more durable, sapwood is less durable. Heartwood gives the tree mechanical strength and support. Sapwood is actively involved in transport of water and minerals from roots to the leaves. Next is the activity of cork cambium. As secondary growth continues, the epidermis ruptures due to increase in thickness or girth. To maintain physiological integrity, the plant develops secondary protective tissues forming the periderm. Periderm is composed of phellem or cork, phellogen or cork cambium, and phelloderm or secondary cortex. Periderm protects the tree from desiccation, mechanical injury, and invasion by pathogens. The phellogen or the cork cambium is a lateral meristem. It is thin-walled, rich in cytoplasm, with prominent nuclei, rectangular in shape, and one or few layers thick. It gives rise to the periderm by periclinal divisions. In stems, the cork cambium may arise from the cortical cells or from the hypodermis. It divides and gives rise to new cells on both sides, cork or phellem on the outer side, and secondary cortex or phelloderm on the inner side. The phellem is multilayered tissue whose cells are dead, impregnated with suberin, and are thick-walled. The cells are rectangular, compactly arranged with no intercellular spaces. As the cork cells are suberized, they are impermeable to water and gases, resistant to pathogens, provides thermal insulation, and prevents excessive water loss or desiccation. Commercial cork is obtained from Quercus suber. The phelloderm or the secondary cortex are made up of thin-walled living parenchyma cells which may contain starch, tannins, or chloroplast. These cells help in the storage of food materials like carbohydrates and fats, and also in healing of wounds. All the dead tissues that lie outside the active cork cambium constitute the bark of the plant. It includes the epidermis, hypodermis, periderm, etc. The outermost dead layered portion of the bark is called retidome. When the cork cambium appears in the form of a complete ring, the bark that is formed peels in circular strips and is known as ring bark. And when the bark comes away in the form of scales, it is known as scale bark. Depending on the texture of the bark, it may be known as smooth bark as seen in beech tree, scaly bark as in eucalyptus tree, or fissured bark with deep cracks as in oak tree. The function of the bark is to give mechanical protection, prevent water loss, prevent invasion by pathogens, and help in storage of metabolites. Thick bark provides resistance to fire and frost. Next are the lenticels. Lenticels are aerating pores formed in the bark. They help in gaseous exchange between the internal tissue and external atmosphere. They are formed beneath the stomata or groups of epidermal cells. They are thin-walled, lens-shaped or elongated or circular cells. The lenticel consists of a loose mass or small complementary cells with numerous intercellular spaces. Lenticels facilitate gas exchange by providing a pathway for oxygen to enter and carbon dioxide to exit out of the tree trunk and thus helping in cellular respiration. Lenticels also contribute to water loss from the plant through lenticular transpiration. Next, let us understand the secondary growth in dicot root. Secondary growth in dicot roots involve formation of vascular cambium from conjunctive tissue located below the phloem and from pericycle cells lying outside the protoxylem. In dicot roots, the vascular bundles are radially arranged. Xylem is exarch with protoxylem towards the periphery and metaxylem towards the center. The cambial cells divide tangentially repeatedly, forming a continuous wavy band of cambium. The cambium divides and forms new cells on the inner side, which later constitute the secondary xylem. It forms the main bulk of the root. The cells are thin-walled, with numerous large vessels, abundant wood parenchyma, and few wood fibers. The cambium ring becomes circular. As more wood is added, the primary phloem is pushed out. Primary xylem is present at the center. The cambium also forms radial bands of parenchyma, which constitute the medullary rays. On the outer side, the cambium divides and forms new cells, which constitute the secondary phloem. It consists of sieve tubes, companion cells, abundant parenchyma, but few bast fibers. The primary phloem gets crushed. Next is the activity of Cork cambium. The single-celled pericycle becomes meristematic and forms thin-walled cells that constitute the Cork cambium or Phellogen. It produces dead, suberized protective cells on the outer side known as cork or phellem, and living parenchyma cells on the inner side forming the secondary cortex or Phelloderm. The phellem, Phellogen, and Phelloderm are together known as the periderm. It increases the girth of the root. Endodermis and cortex get disorganized. Finally, let us look into the differences between secondary growth in dicot stem and dicot root. The cambial ring formed in dicot stem is partially primary and partially secondary in origin. In dicot root, the cambial ring is secondary in origin. In dicot stem, the intrafascicular cambium and interfascicular cambium form the vascular cambium. In dicot root, the conjunctive tissue and the pericycle form the vascular cambium. The cambial ring is circular in dicot stem, while in dicot root, it is initially wavy and later becomes circular. Periderm in dicot stem originates from the cortical cells. In dicot root, periderm originates from the pericycle. In dicot stem, more amount of cork is produced, while the cork produced is less in dicot root. Lenticels on periderm are very prominent in dicot stem, while they are not prominent in dicot root. So today, we have learnt in detail about the secondary growth in dicot stem and dicot root with their differences, formation of annual ring, activity of vascular cambium and cork cambium, differences between springwood and autumnwood, heartwood and sapwood. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.